Hello and welcome to Opponent Process. This will be the first of 3 presentations that talk about opponent process. This first one will be color vision, the next one will be neurons, and the next one after that, the last one will be about hunger, if I remember correctly. I'm not going to read each one of these slides, so pause and rewind will be your friend. I just want to note at the very bottom of this that although the information that I provide should be accurate, that it's not going to be all-encompassing. For example, for color vision, there are actually 3 different kinds of theories that govern color vision, and the one we're going to be talking about by Ewald Helling is actually only one of them. So let's begin. This is a rough representation of the eye with part of the eye cut away. This is the part that light enters into, and this is the back part. This part is the optic nerve. This part in the back is called the macula. It's where most of your photoreceptors are, and this part inside is the fovea, which we'll talk about in a moment. The macula is where most of the rods and cones are. I guess you could say that this is where your 20/20 vision comes from. Where there are some rods in the outside of the macula, not a lot. Most of your vision is here. So let's go on. The fovea. This is where your most accurate vision is. This is the part where if you were to join your thumb and forefinger together and put your arm out all the way with this OK sign, then look through the OK sign, that's about the size of your most accurate vision at a distance. You might say, but my vision is very clear all over the place, not just in that small area. And you would be right. But your eye can only see a small amount of very detailed area at any one time. The thing is that you have 2 eyes, and both of them are darting all over the place constantly. So the area that apparently looks clear is actually much larger than the area that is actually clear. Let's see, hang on a second. All right, this next slide is further inside the eye. This is not entirely accurate. It makes it look like there are only rods here. There are actually cones there too, but I wanted just to point out that the fovea is where most of the action is happening, where your most detailed vision is. So what does this all have to do with opponent process? These are 2 particular cones. These are 2 different colors in the same cone. You have many of these cones in the back of your eye, but not, I don't know how to say this. If one is registering green, that particular cone cannot also notice red. This one, let's say, is noticing green, and this one, let's say, is noticing blue, but another red-green cone that's next to it might be registering red if there is red in the environment. So there's something in the environment that has green in it that fires the green side of the cone. That impulse goes down the optic nerve and registers in your brain, and you apparently see green. But notice that the other side is not really working. This side is, this side is not. Right? But as you look at something green, in this case, for a longer and longer period of time, that side of the receptor actually gets fatigued. It will get tired, just like a jogger gets tired. So this side is working, and this side is not working at all. But a funny thing happens when this side actually stops working. And what happens is it fires the opposite side in order to chemically balance the cone. So this side is actually turned off, it's fallen asleep, so to speak. And this side has turned on, even if, and this is important, even if there's nothing red in the environment. So if you were to stare at something green, until that particular receptor turned off. When you looked away at a blank space, this side would be firing. So you might see spots or a shape in the color red, even though there's nothing there. This is what we would call an after image. So this may sound kind of unbelievable. Well, what do you mean if I look at something for long enough that a cone will turn off? That doesn't make any sense at all. So although, because of the way Screencast-O-Matic works, this is not quite what I wanted to show you. Here is a box with colored dots in it. What I want you to do is stare at the little plus sign in the center. Stare and try not to blink, try not to move your eye around too much, just stare at it. So let's stare together, shall we? All right. You can use pause, or you can go back to this later if you need to. What should have happened is at least some of the dots should have disappeared completely. Now, if you blinked or if you moved your eyes around a lot, those that disappeared will come back again. But some of them hopefully disappeared. That is opponent process. Now, if you'd looked away at a blank space, you might have actually seen whatever the opposite color is for that. But for the most part, you should have seen some dots disappear. This is probably not going to work because I don't think I made the colors distinct enough. But technically, if you looked at this image where yellow, green, and black are, that if you stared at this for long enough to get the opponent process working and get black to turn off into white, get green to turn off and turn into red, and yellow to turn off and turn into blue, then this is actually what you would see if you looked away. Now, not that you looked away at a flag that was flying in the wind, but you looked away at a blank space. So you start with this, and you end up with that. So, how does this apply to opponent process? When one part of color vision is activated, the other part is not activated. It is inhibited. So that's really all that opponent process is. When one part is on, the other part is off. They can't both be on at the same time. Please notice dichroma and achroma. Achroma means no color, and dichroma means two. Now, if I understand this concept correctly, this is also a way that motion works. You can actually stare at something for a while if it's moving, say like a train going by, and that when the train disappears, it will seem as though things in the background are moving in the opposite direction because your sense of motion has become fatigued. Anyway, I hope you've liked this. I droned on for a little while. Please go to the next part about neurons. Hello, and welcome to Opponent Process Part 2. In this part, we'll be talking about the nervous system. In particular, we'll be talking about the everything else. The CNS has to do with the brain and the spinal cord. The eyes aren't really included, but I just wanted to draw them for effect. What we're really going to be talking about, for the most part, is the autonomic nervous system, which is part of the everything else that you saw in the last drawing. The sympathetic nervous system is the one that activates your body. It's ready to do something. It's either really, really happy and excited and running around, or it's afraid, and running around is more like running away. Here in the parasympathetic nervous system, it's designed to do the opposite, to calm you down. Perhaps listening to relaxing music, you are more calm than you were before. But you can't be afraid and calm at the same time. These are some things that the sympathetic nervous system does. It could be in reaction to fear. It could be in reaction to something good that you're really excited about. Perhaps it's a birthday. graduating from high school, getting your driver's license, something like this. The parasympathetic does the complete opposite. Whatever was in the last slide, the parasympathetic does the opposite. This is Bob. Bob is a slightly overweight college freshman. Excuse me. And he's very afraid of spiders. I know. Let's show one. Bob didn't like that very much. This is what we would think of as emotion tied to the sympathetic and the parasympathetic nervous system. Bob got his sympathetic nervous system activated. He was afraid of spiders, he got activated, and now there are two ways for him to not be afraid. The first part is time passing, right? Is that the neurotransmitters are just metabolized. You just wait for that emotion to pass. But it can actually be short-circuited by you thinking. So this is the time passes. Oh my gosh, there's a spider. Okay, wait, it didn't do anything. Oh, I'm fine. Right? Or we show Bob the spider. But then Bob has the idea. Oh, that's just a spider. It's really not going to hurt me, and probably, even if it really wants to, its fangs are too small to penetrate my skin. A much shorter pathway to turning off the sympathetic nervous system. Now we're going to talk about neurons and the more mechanical biology. Here you see neurons with, although you can't really tell which one is which, they have dendrites, they have a lot of dendrites, which receive signals from other neurons, and they have one axon which sends signals from that particular neuron to its next adjacent neuron. Here we can see what looks like a little clip from a film showing neuron transmission down the dendrite to the cell body. But this is actually where most of the magic actually occurs. It looks almost like two pieces of something. Oh, there was one piece and they both got ripped apart. But in fact, what it's trying to show is neuron transmission between an axon and a dendrite. This is the end of an axon. Notice there are vesicles. Each one of these vesicles is probably not filled with the same neurotransmitter. Whatever happened to that original neuron, this green one, is that, let's say it's an excitatory signal, meaning get excited, something is happening. One may be one neurotransmitter for excitement, the other one may be something completely different, and the third one something completely different after that. Then the vesicle closes up and actually retreats back up through the axon. Here we can see the dendrite, which has receptor sites on its surface. So what happens then is that neurotransmitter is released out of these vesicles and these neurotransmitter pieces fly towards the dendrite. The brain actually is very economical. If not all of these neurotransmitters are used, they'll actually be sucked back up into the axon to be used later. So the brain is not wasteful. What happens then when one of these neurotransmitter pieces falls to or flies towards the dendrite? And that's what you see here. Please notice that not all of the receptor sites are the same. Hence, that's what I was trying to show with the different colors, is that more than one kind of neurotransmitter is sent to the dendrite at any one time. What happens then when a neurotransmitter reaches the appropriate receptor site? Like here, we have this green one that's round. And this is actually what happens. The receptor binds to the neurotransmitter and it is sucked down into the dendrite. Let's say this is an excitatory, like I can't think of one at the moment. Adrenaline, let's say, that it binds here, goes down into the neuron, And that neuron then passes on down its axon an excitatory signal. What happens if a neurotransmitter tries to bind with a receptor site that it's not designed for? And the answer is nothing, really. It is sucked back up into the vesicle, as I showed you earlier. So it just bounces off pretty much. Here, what you see is an inhibitory. This is let's say this is DBI, which is diazepam binding inhibitor. This is a diazepam binding or docking receptor site, but this is not diazepam. This is something else. This is something that's blocking it. So diazepam calms you. That means little things will be coming and bouncing right off. Bounce, 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 bounce. What was I talking about again? All right, sorry. So notice that there is no signal going down into the dendrite because this is blocking it. So, opponent process. When a neuron is excited, it cannot be calm at the same time. When you feel an emotion, you cannot be happy and, let's say, excited and calm at the same time. One has to pass for you to have the other. Now, there is a way for you to be so happy that you start to cry, and we can talk about that later, but that's not part of this particular essay, or you don't know what I'm talking about, PowerPoint. A response can be triggered or it can be inhibited. What's interesting here is that whether a neuron is being excited or inhibited, now not blocked, but inhibited, like a transmission that says, don't do that, the neuron is actually still working. There isn't hardly any time that I can think of when the neuron is actually not doing something. It's either sending signals saying, do that, do that, do that, or it's sending signals saying, don't do that, don't do that. But that's opponent process. It's either on or it's off. It's excited or it's inhibited. Thank you. The next PowerPoint will be about hunger. Hello, and welcome to the last part of opponent process. In this particular part, we'll be talking about eating behavior. As you can see on the screen, these things have to do with the brain, the stomach and the small intestine, and the blood itself. Eating is an animal behavior, but in this particular case, we're talking about human behavior. So as you can see, we have Bob back again, the infamous person who was afraid of spiders. Inside of Bob's head is his brain. And inside the core of the brain is the hypothalamus. The hypothalamus is not just one part. It's actually a couple of different parts, but the two that we'll be talking about today, or in this particular part, is the lateral and the ventromedial hypothalamus. The lateral hypothalamus is what starts eating behavior. It's the thing that makes you understand that you are hungry. The hypothalamus itself, as an organ, monitors levels in the blood. Levels of what? Levels of hormone, levels of blood glucose, and other things. But in particular, the lateral hypothalamus is what starts eating behavior. And primarily it's set off by low glucose levels in the blood. The ventromedial is actually what stops eating behavior. A chemical called CCK, which we'll talk about in a moment, is the chemical that signals to the hypothalamus that you are full, really full. Now we're not talking about appetite. This is important. Appetite is your desire to eat. We're not talking about desire to eat. We're actually talking about if you don't eat, you're going to pass out, or you may feel sick. But CCK is the chemical that is released into the bloodstream. It actually takes about 20 minutes or so for CCK to get to the brain. Hence, your stomach could be full, but you wouldn't necessarily know it. Now I don't mean full like to the point where it's going to break, but full to where you don't need to eat anymore. So when you do eat, you should eat slowly. So here Bob realizes that he needs food. He doesn't feel well. He maybe feels a little tired. His stomach may be growling. He may feel a little dizzy or lightheaded. Here is the organ that is the repository for your food. Now what's important is, and I think I wrote this, is digestion starts with the mouth. You mechanically break down food in your mouth. It is chemically broken down here in the stomach. So once it's in the stomach, you can see this is the custom crushed velvet stomach. Inside the stomach, the breakdown of food continues. You have stomach acid. Anyway, blah, blah, blah. Here we go. That during times of extreme stress or hunger, that the stomach, which is a muscular organ, actually starts to contract and sort of move. Now, just because your stomach is growling does not mean necessarily that you're hungry. It could actually be just that the stomach is moving, and we don't really know all the reasons, all the conditions under which the stomach does contract, but that you don't have to be hungry in order to hear your stomach moving. But when you are hungry, the stomach does move. This kind of shows a stress reaction where the stomach acid is rising and the stomach is moving also. So Bob is hungry. He realizes he needs to eat food. Donuts. All right, I hope this isn't during the lunch period. So the stomach is about the size of your fist, or approximately. It's pretty small, and the amount of food that the average person eats will stretch the stomach so that it's a little larger than that fist size. When it starts to stretch, and when sugars begin to be sensed by I think it's called the duodenum, I can't remember, but the first couple inches of the small intestine, those two things, the stretching and the sensing of sugars in the small intestine, release CCK, which is at the bottom. You don't have to know the whole word. I'm only going to refer to it as CCK, both here in this video and on the test. CCK then goes, the problem is that it doesn't just go from the stomach and then like from some nerve impulse goes directly to the brain. It doesn't work like that. It actually goes from the stomach into the bloodstream, and then it's called a systemic. It has to go all over the whole body, and eventually it gets to the brain. But it takes about 20 minutes for that to happen. So, opponent process deals with eating behavior because you cannot be hungry and satisfied at the same time. You cannot feel full and feel hungry at the same time. However, again, this does not have to do with appetite. Appetite is your desire to eat. You may not feel hungry, but you may look at something, and then decide, ooh, one of those would feel so nice. I would gladly play you Tuesday for a donut today. All right, so then what happens is that you feel full and CCK, after CCK has been released. And I think that's about it. This is actually the shortest video of all three. But upon process, if you have any questions about these, you can email me at the high school website, or you can talk to me in class. Thank you, and I hope these were informative.